Hi all, and welcome to the first session of Regents Review. I am Miss Winans and I'm here to go through experimental design. Okay, the first task for the Living Environment Regents that we're going to cover today is the design of an experiment. In middle school, you went over the scientific method, and most likely your teacher had you memorize the steps of the scientific method. For the Living Environment Regents, you have to take it one step further, and that step that you're going to take is you need to be able to understand the components of an experiment and be able to design an experiment of your own. The way you're going to experience this the day of the Regents is you're going to be given a passage, and that passage is going to give you information about a specific problem that scientists are researching. They may design the experiment for you, and then you need to identify the components, or you may have to design an experiment completely on your own. So if you look at this slide, you still see the components of the scientific method. You see identify the problem, research, hypothesis, design, perform experiment, collect and analyze data, state a conclusion, repeat, share, and review. Again, you're not going to be asked to memorize these steps. You're going to be asked to apply them. So based on the question, it's going to give you a passage. It's going to tell you about a particular experiment that a scientist performed, or a particular experiment that a scientist wants to perform. And that is going to allow you to identify the problem and research. Based on your reading, whatever the experiment is about, whatever the question is about, make sure you underline that the day of the test, because you need to establish a hypothesis based on what the question is asking you. You should recall that a hypothesis is a statement about the experiment that identifies how you will manipulate that experiment, which we call the independent variable, and what you expect to happen, which is measured by the dependent variable. Therefore, when you write a hypothesis, you are going to write it as an if-then statement, where the if statement identifies the independent variable and the then statement identifies the dependent variable. Like I said before, the day of the test, you will either have a reading passage in which you are going to have to identify the components of an experiment, or you will have to design your own experiment. Either way, you have to be well versed in your ability to design an experiment. The first thing you need to know are the variables in an experiment. A variable is a component of an experiment. It's part of your experiment. The three variables that you need to identify are the independent variable, the IV, the dependent variable, the DV, and the constants. The independent variable is what I, the experimenter, will do to manipulate the experiment. So how am I changing the experiment? The dependent variable, D for data. What type of data am I going to collect? How am I going to measure how this change has affected my experimental results? The constants are going to be variables that are found the same in both groups. The groups in an experiment are called the control group and the experimental group. The control group is the group that's under normal conditions, standard conditions. In other words, the control group will not contain the manipulative variable. It will not contain the independent variable. The experimental group will be virtually identical to the control group with one exception. You will add the independent variable. It's very important that you have a control group and an experimental group in an experiment. You can have a control group and multiple experimental groups, but you must have at least one control group and one experimental group. This allows you to compare the results. It provides you with evidence. Remember, the control group is not manipulated. Since it's not manipulated, you can use the control group to prove that it was the independent variable in the experimental group that caused the change in results. In order to prove that your experimental results are valid, you or other experimenters should be able to repeat the experiment and get the same results. The more times you do an experiment and get the same results, the more evidence you have to support that your independent variable changed the experimental results or did not change the experimental results. So repetition, repeating the experiment, doing the experiment over and over again, will make your experimental results more valid. You can do this two ways. You can repeat the experiment over and over and over again, or you can increase your sample size. In other words, if you have one plant in your control group and one plant in your experimental group, you're only doing the experiment once. But if you have 50 plants in your control group and 50 plants in your experimental group, you increase the sample size and you actually perform the experiment 50 times during that one experimental round. Okay, let's review a couple components to a well-designed experiment. First question. An experiment should contain control and experimental groups. Why? Take this time to jot down a couple reasons why an experiment would need a control and experimental group. For comparison, you always want to have a control group and experimental group because the control group and experimental groups are virtually identical, except for the fact that the experimental group contains the independent variable. By having a control group, you have evidence that the change only occurred in the experimental group, meaning the independent variable caused that change. Without the control group, you wouldn't have evidence and you wouldn't have the ability to compare the effects of the presence of the independent variable versus the absence of the independent variable. Next question. Why should an experiment contain a large sample size? Remember, repetition increases the validity of your experiment. The more times you or another scientist can perform your experiment and get the same results, the more evidence you have supporting your hypothesis. Large sample sizes allows you to do multiple trials of the experiment within one round of the experiment. This allows you to have more evidence to support or reject your hypothesis. Next question. Why should only one independent variable be used during an experiment? Remember, your control and experimental group should be virtually identical to each other except for one factor. That factor is the independent variable, how you are manipulating the experiment. If you manipulated more than one thing, you wouldn't know what caused the change. For example, if you were testing the effects of light on plant growth, you would probably put 10 plants in the dark and 10 plants in the light. You need to give them the same amount of soil, same species of plant, same amount of water. If you were to change any of these other factors, you wouldn't be able to say which factor caused the change. Okay, so let's take a look at this question. The study effects of common substances on the heart rate. So we're trying to see the effects of heart rate. Let me just get a color here. Heart rate on a tiny aquatic. Those of us who don't know what aquatic means, it means that it lives in water. Organism known as Daphnia. Students placed a Daphnia in a drop of water on a glass slide. The students then added 1 or more drops of a test substance dissolved in water. So that's something that we want to pay attention to because it looks like the students are adding something else to the experiment. They waited 10 seconds, then counted heartbeats for 10 seconds. So they added a substance and then they counted heartbeats. The students used a clean slide and a new Daphnia each time. Their data table is shown below. First thing we have to remember is whenever we make a data table, the data table will always have a title, and then you will always put your independent variable on the right side of the data table and your dependent variable on the left side of the table. Knowing this trick, it's going to help us identify these variables if we couldn't figure it out from the reading. So according to my key, the independent variable should be the test substance, and the dependent variable should be the hearts per minute. So if I go back to the reading, I remembered that the independent variable is how the scientist or the experimenter manipulates the experiment. And lo and behold, we see that the students added 1 or more drops of the test substance. We also remember the trick with the data table that IV goes on the right, DV goes on the left. So the independent variable in this experiment, the IV, is the test substance or the coffee and ethanol, right? Because that's what the experimenter did to the experiment. That's what they added, okay? The dependent variable, the data, is how we are going to control or keep track of how the independent variable affects the substance or organism that we're testing. So the way we're going to figure out if coffee or ethanol has an effect on the Daphnia is we're going to count the heartbeats. We also could use our trick over here. And remember, IV goes on the right, DV goes on the left. So our dependent variable is heart rate. Okay. Identify the experimental groups in the experiment. So the groups containing the independent variable are going to be the experimental group. So that's going to be the coffee and the ethanol. Propose a liquid that could be used for the control group. So remember, we went back to this term that we may not know, and I said that aquatic means that the organism lives in a watery environment. So I'm going to propose that water would be a good control substance, a good liquid for the control group. So the last one, it says evaluate how the instructions could be changed to improve the design of the experiment. And whenever you want to improve an experiment or make an experiment more valid, you just increase the trials. So you would either repeat the experiment or you could increase the sample size, which means that you could increase the amount of Daphnia in each group. I hope that helped. Okay, let's try this next question. So here is the Daphnia that we were looking at in the previous experiment. You can notice that we're emphasizing the heart. The Daphnia is transparent, which allows us to see the beating of the heart. We can also see the beating of the legs down below, but we're just going to focus on the heartbeat for now. So the question says, Daphnia are freshwater organisms sometimes referred to as water fleas. Design an experiment that could be used to test the effects of temperature on the size of a Daphnia population. So I'm going to get my pen out and I'm going to underline exactly what they want us to test. And they want us to test the effects of temperature on the size of the Daphnia population. So this is different than the previous one because in the previous sample we were testing the effects of a substance on heart rate. Now we're testing the effects of temperature on the size of the population. So what we're talking about is the amount of Daphnia. So how many Daphnia we have alive or how many Daphnia we have dead. So what I'd like you to do is take a little bit of time and think about this. I'd like you to write down your hypothesis, describe how your control group will be tested differently from your experimental group, identify the independent variable in the experiment, and identify the type of data that will be collected during the experiment. Okay, so what we should have done here is, again, we are designing an experiment to test the effects of temperature on the size of the Daphnia population. Okay, so the first thing they asked us to do is a hypothesis. Hypothesis has to be an if-then statement, and it has to be based on what the question is asking us. So I'm going to increase the temperature. So if temperature increases, then the Daphnia population will decrease. Okay, the next thing they wanted us to do was to identify the variables, I think, right? No, okay, so they want us to test to see if what goes in the control group and what goes in the experimental group. So in my control group, I'm probably going to put Daphnia. I'm going to be specific, so I'm going to put about 20 Daphnia. They live in the water, so I'm definitely going to need water. I can't pour this on the table, so I'm definitely going to need a tank to hold that water. And 20 Daphnia, I'm going to use a thermometer. To measure the temperature, and I'm going to keep this at room temperature. Okay, which is about 30 degrees Celsius. Okay, and then in my experimental group, I'm going to put all of this stuff into my experimental group, except the temperature I'm going to increase to boiling, which is 100 degrees Celsius. Okay, so all of these factors that are in both groups, those are my constant variables. So if they asked me to identify some of the constants, I could have put 20 Daphnia, the water, the tank, the thermometer. Okay. All right, the independent variable is going to be how I manipulate the experimental group. So I changed the experimental group by increasing the temperature. So the IV in this experiment is temperature. Okay, the dependent variable is how I measure the effects. So again, remember that one trick is if. The if statement tells me my IV, and the then statement tells me my DV. So the Daphnia population, so I'm going to count the number of Daphnia deaths. Okay, you can also be more positive and you can count the amount of Daphnia that survive the 100 degree Celsius. The last question is identify the type of data that will be collected. That is going to be your dependent variable. And I hope this helps. Okay, another type of question that you will be asked to answer on the Living Environment Regents is a graphing question. During the graphing question, you will be asked to make a proper scale, label the axes, make sure the independent goes on the x-axis and the dependent variable goes on the y-axis. You are going to take an existing data table and you're going to plot the points and notice the trends that the data indicates. So for this example, we're going to go back to our friend the Daphnia. Daphnia are used as a model for heart rate experiments because the Daphnia are transparent, and as you can see in our model here, we can see the beating of the heart. So we can change different environmental factors and see if they increase or decrease the heart rate. So the question reads, 3 students each added equal volumes of pond water to 4 beakers and placed each beaker in a different water bath. Each student maintained the water baths at temperatures shown in the data table. The students then added an equal number of water fleas to each of their 4 beakers. After 1 hour, the students used microscopes to determine the average heart rate of the water fleas. So we're paying attention to the heartbeat. The procedure was repeated for a total of 3 trials at each temperature. So we're also dealing with changing temperature. The results of this investigation are summarized in the table below. So again, when we're looking at a data table, the IV is on the left, the DV is on the right. When we do our graph, now we're going to put our independent variable on the x-axis and we're going to put our dependent variable on the y-axis. So basically what they want us to do is they want us to draw a graph that shows the effects of water temperature on the heart rate of the Daphnia. The other thing we want to pay attention to closely are the instructions they give you right here. So they're telling us as we plot the points that we should be surrounding each point with a circle. If you don't put a circle around the data point, you will lose 1 point on your Regents question. So just make sure you're following directions as well. When graphing, there are some rules that you should follow. The first rule is that you need to make sure that you draw the graph to scale. And the second rule is that you need to declare your starting point for your x and your y axis. I find the easiest way to do this is to put, I'm going to use the color blue so we can see this a little bit better, is to put a little 7 here. And this way you can start your graph at any point that you want. So if you wanted to start at 5 on the x and 0 on the y, that's perfectly fine. So it allows you to spread your graph out more and it may help you with your scale. But I'm actually, for this graph, I'm going to start the x and the y at 0, but I'm going to declare a value for both because that makes the person who's looking at my graph understand exactly what my starting point is. So the next thing I'm going to look at is I'm going to look at the values on my dependent variable that are going on the y and the values that are on my independent variable, which is going on the x, to figure out a scale. So for water temperature, I am going from 5 to 35. So it looks like I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 boxes at the bottom. So I think I'm going to go up by 5s, but I'm going to go every 2 boxes. So 5, 10, 15, 20, 25, 30, 35. Now I'm also having a little difficulty figuring out where my boxes are, so I'm going to draw these little lines down to help me out because I keep on getting stuck when I'm looking at it. So just to make sure I'm going every 2 boxes. Okay, and 40. All right, so I have a little more of a challenge on the other side because I have to go all the way up to 280. So I'm going to give it a try, and I'm going to maybe go up by 20s. I draw those lines again because I was having a lot of difficulty making sure I was going up by 2 boxes. 1, 2. Now, it's very important that you do your graph in pencil because sometimes you might think that you have an awesome scale and then you get all the way to the top and you remember that, oops, or you see that you don't have enough boxes. And what I've seen some pretty interesting things done on the day of the Regents. Sometimes students will add extra boxes to their graph paper, but if you do it in pen, you're kind of out of luck. So always do a graph in pencil. This allows you to have the ability to erase your scale if it doesn't fit, and you definitely cannot do what those students do and draw extra graph paper because then you'll lose all credit for your graph. Okay, so it looks like my scale fits, so that's good. The next thing you're going to do is plot the points. So what you're going to do is, as you plot the points, you're going to start with the first one. So we're going to go 5 to 40. So 5, 40. And remember those instructions, they said to put a circle around your data point. So make sure you put a circle there. Okay, so you continue to plot the points. After you plot the points, you then connect the dots, and then you should be able to successfully see the trend in your data. Okay, so we're going to end this review session with 2 additional practice questions. One will be experimental design, and the last one will be a graphing question. So this question states, scientists have been experimenting with different forms of alternative energy to help reduce the amount of fossil fuels that are burned. They studied yeast, which convert plant materials into ethanol, a form of alcohol that can be used in automobiles. These experiments were carried out at room temperature. The scientists wondered whether more ethanol would produce at different temperatures. So I'm going to underline a couple things. So it seems like they have something about room temperature. And then, so here's the big thing. This is what the scientists are wondering. And then what they're wondering is that, is more ethanol produced at different temperatures? So based on this reading, I think that I'm going to be measuring ethanol and changing the temperature. It says your job is to design an experiment to determine the effects of temperature on ethanol production. Okay, so we're going to change the temperature again and measure the ethanol production. Okay, so take some time, state your hypothesis, and identify the items in the control group, the items in the experimental group, 2 constants, the independent variable, and the dependent variable. For those of you who have difficulty with the experimental design questions, experimental brainstorming will really help you understand how you're going to set up the experiment. So if you do this brainstorming before you answer the actual Regents question, you'll have a better way or reference to refer to when answering the bullet questions. So again, we're dealing with the ethanol production at different temperatures. So it looks like we're going to be measuring ethanol and we're going to be changing the temperature. So from this, I'm going to pull that this most likely, if I'm measuring it, that's going to be the data I collect during the lab. So that's going to be my DV. And I'm changing the temperature, so that's going to be my IV. So the first thing you want to do in the experiment is an if-then statement. The IV is in the if statement, the DV is in the then statement. So if the temperature is increased, and I'm running out of room, so I'm just going to draw an arrow up, comma, then I said the dependent variable would be the ethanol. So then more ethanol will be produced. Okay, so I have my hypothesis. So the next thing I probably want to figure out is what I'm going to put in my control group. So let's go back to the reading. The first thing I want to figure out is who is producing the ethanol, or what is producing the ethanol. And it looks like in the reading it's yeast. So I definitely have to add yeast to my experiment. I'm going to go back to the reading and see what these yeasts need to produce ethanol. And here in the line they said that they convert plant materials into ethanol. So it looks like I'm also going to need some plant materials. And since it's temperature, the standard conditions would be room temperature, which we said in the previous example is 30 degrees. Okay, so if I take a look at this, my experimental group, and I think I'm going to do 3 groups. Okay, they're going to contain all the constants. Okay, so the constants are going to be the things that are found in the same group. So this is going to be constant. Now the thing that I'm going to change, this is not going to be constant, is the temperature. And why is that? Because it says it right there in the reading. So since I decided to do 3 experimental groups, I'm just going to steadily increase the temperature. So 40 degrees Celsius in group 2, I'm going to do maybe 50 degrees Celsius. Group 3, 60 degrees Celsius. Okay, so I know at least 2 constants, yeast and plant materials. I know that the thing that I'm changing, the one difference between my control group and my experimental group, seems to be temperature. So this is my IV. And I'm going to go back to my dependent variable for my dV. In my hypothesis, that's going to be the ethanol produced. And remember, all of this information I did not make up on my own. All of this information came directly from the reading. So whenever you're doing a Regents question, you have to make sure that all of your answers are coming from the reading. So after you brainstorm the setup of the experiment, you should be able to try the Regents question, and that's on the next slide. So give it a try.